In the stillness of a summer clearing, when the air hangs warm and heavy between leaves that hardly stir, a wasp drifts into frame with the kind of mechanical precision that evolution tends to perfect only through relentless trial. Its wings carve thin vibrations through the air, producing a faint tremor more felt than heard. But it is not the wings that command the camera's attention. It is the weapon tucked beneath the wasp's abdomen, a weapon so small it disappears if the light shifts even slightly, yet so refined that it can change the fate of aperture many times the wasp's size. In a magnified view, the stinger reveals itself not as a simple needle, but as a living instrument of engineering. It is a modified ovipositor, the ancestral structure once used solely for placing eggs into soil or host tissue. Over millions of years, selection pressures reshaped it into a surgical tool capable of slicing, anchoring, and delivering chemical messages that the nervous systems of other animals cannot ignore. At extreme magnification, the stinger splits into two sliding lancets, each tipped with minute barbs arranged like backward-facing hooks. These barbs do not simply pierce, they secure. Each time the wasp pulses its abdomen, the lancets alternate, ratcheting the stinger deeper into flesh without the need for force. It is a mechanism so efficient that the camera must slow the moment dramatically for the movement to be visible at all. As the stinger advances, the biological choreography has only begun. Inside the slender channel between the twin lancets, venom travels like a dark current. One species uses a venom designed to collapse nerve membranes, preventing muscles from receiving the electrical signals that command them. Another wields a chemical mixture that disrupts blood physiology, causing tissues to swell, clot, or dissolve, depending on what the wasp requires. But perhaps most unnerving are the venoms that do not aim to kill or paralyze in a traditional sense. Instead, they alter behavior, dismantling instinct from the inside, leaving the victim alive but compliant, a functioning body with its evolutionary drives temporarily silenced. A slow-motion sting on a spider captures the moment when this transformation begins. The spider rears back, legs raised in warning, but the wasp is faster by orders of magnitude. Its stinger finds the precise gap between exoskeletal plates where the nervous system lies closest to the surface, and with a single measured thrust, it reaches the neural hub that coordinates movement. The venom acts within seconds. The spider's limbs lose their rigidity, folding like tent poles without tension. Yet the spider remains aware, its eyes tracking movement, its body intact. Only the connection between intention and motion has been severed. The wasp steps back, antennae sweeping in calm, calculated arcs, as the spider tests its limbs and finds them unresponsive. Evolution has crafted this moment with meticulous purpose. In parasitic wasps, the story grows more complex. The camera follows a smaller species, barely noticeable until it climbs onto the metallic green thorax of a cockroach. What appears to be an ordinary sting is, in reality, a two-part neurological operation. The first sting targets the thoracic ganglia, clusters of nerve cells that command leg motion. This dose temporarily paralyzes the roach, preventing escape. But the second sting is far more precise. The wasp guides its stinger directly into the roach's brain, reaching the region responsible for initiating locomotion. The venom injected here does not destroy tissue. Instead, it alters neurotransmitter activity, effectively removing the internal impulse to flee. The roach can walk, run, or struggle if pushed, but it lacks the instinct to initiate these actions on its own. It becomes a body without urgency, a creature whose natural responses have been gently disconnected. The wasp then performs what biologists sometimes call behavioral hijacking. It nips the roach's antenna, not to injure, but to lead. The roach follows the tug like a docile animal on a leash, stepping forward with obedient slowness as the wasp guides it into a burrow. There, still alive, it becomes a living food chamber for the wasp's developing larva. The larva will feed for days or weeks, consuming tissues in a strategic order that keeps the host alive as long as possible. This is not cruelty in a human sense. It is an evolutionary contract, refined through uncountable generations, where efficiency determines survival and emotional considerations do not exist. In another scene, a different parasitic wasp approaches an orb-weaving spider suspended in its web. This species does not simply immobilize its host, it reprograms behavior entirely. A single sting delivers compounds that influence the spider's neural circuits responsible for web construction. Over the next day, the spider begins weaving a structure unlike any it has ever built, a reinforced web with fibers arranged to maximize stability rather than prey capture. This web is designed for one purpose: to cradle the wasp's cocoon. Once the altered web is complete, the spider settles into it, no longer resisting. The wasp returns, deposits a single egg, and then leaves the spider alive, but near the end of its usefulness. The larva will eventually feed on the spider, using the specially built web as a safe cradle suspended beyond reach of ground predators. The spider, having unknowingly completed its final task, succumbs shortly after. What makes these behaviors even more remarkable is the minuscule quantity of venom required. The amount delivered by a wasp stinger is often measured in micrograms, such a small mass that it borders on the conceptual. Yet that tiny chemical signature is enough to shut down muscles, change essential motor patterns, or overwrite deeply ingrained evolutionary instincts. Venoms are not composed of a single toxic ingredient, but rather sophisticated cocktails. Peptides that interfere with electrical signaling, enzymes that rearrange cellular machinery, and small molecules crafted to bypass biological defenses. Many of these compounds act with surgical specificity, targeting only the systems the wasp needs to manipulate. The result is a form of biological control so advanced that human neuroscience still struggles to replicate its precision. Even more intriguing is the diversity within venom strategies. Some hunting wasps evolve toxins designed for rapid immobilization to prevent prey from damaging delicate larvae. Others refine slower-acting venoms that preserve the prey's tissues in fresh condition for extended periods. Behavioral venoms, like those used on cockroaches, reveal evolutionary pathways where controlling the host is more useful than disabling it. Each strategy reflects a distinct ecological niche and a finely tuned relationship between predator and prey. As the camera pulls back to reveal the wasp cleaning its stinger, a ritualized motion performed after nearly every sting, the complexity of this evolutionary weapon becomes even clearer. The stinger is not simply a puncturing device, it is a delivery system, a sensory probe, and a precision tool guided by instinctual knowledge of anatomy. With it, the wasp navigates the fragile line between life and death, control and chaos, and survival and extinction. In this miniature world, power is not measured in size or strength, but in biochemical mastery and the ability to rewrite natural behavior with a drop of venom too small for the naked eye to see. The meadow looks peaceful from a distance, a quilt of tall grasses shifting under a warm breeze that barely bends their slender stems. But inside this green labyrinth, just a few centimeters above the soil, tension hides beneath the calm. The camera drops low, slipping between leaf blades, until the view narrows into a world measured not in meters, but in millimeters. Here, threads gleam like drawn glass, catching sunlight in fractured lines. The structure at the center is unmistakable to anyone who knows the architecture of European fields. The orb web of the wasp spider. Its builder waits nearby, her abdomen striped in bold bands of yellow, white, and black. A warning palette evolved to discourage the careless and the curious. Her web is not a random mesh, but a calculated design, tuned to the physics of struggling prey. Across its center lies the stabilimentum, a zigzag ribbon of densely layered silk that flashes white, even in low light. Whether it evolved as camouflage, structural reinforcement, or an optical lure remains debated, but its function becomes clear when a sudden movement shatters the quiet. A grasshopper, flushed from its routine climb along a blade of grass, leaps without looking, and lands in the center of the stabilimentum with a soft, nearly inaudible thud. From this point, the camera slows dramatically. The grasshopper's legs kick, each movement deforming the silk into trembling waves. Orb webs are energy sensors, converting motion into information, and the vibrating strands transmit a near-instant message across the structure. In the corner of the web, the wasp spider reacts before the grasshopper can complete its second kick. She rushes forward with astonishing speed for a creature of her size, each step anchored by specially adapted claws that grip even the slickest threads. The grasshopper struggles harder, scraping and twisting as it tries to free itself. Its hind legs, powerful enough to launch it many times its own body length, are useless here. Every attempt only anchors it deeper, the web's radial and spiral threads sticking to joints and wing edges. It still has air, though, a fact the spider is about to remove. The wasp spider makes her first contact not with a bite, but with silk. She pivots around the trapped grasshopper while releasing a continuous stream of wrapping threads from her spinning organs. These organs, called spinnerets, each house multiple spigots, microscopic nozzles that draw liquid silk proteins from internal glands and instantly solidify them when exposed to air. But not all silk is the same. The wrapping silk she uses in this moment is coarse, strong, and slightly elastic, designed specifically for immobilizing prey. She swings her abdomen in rhythmic arcs, binding the grasshopper's thorax, legs, and wings in a cocoon that tightens with every circuit. Within seconds, counted now not in minutes but in heartbeats, the prey's limbs are locked together so firmly that they cannot even tremble. Silk under tension exerts pressure on anything inside it. As the layers accumulate, that pressure becomes suffocating. Insects breathe through spiracles, small openings along the sides of their bodies that connect to internal air tubes. When these spiracles are compressed or blocked, oxygen intake drops sharply. The grasshopper begins to fade before venom ever touches it. Only after the prey's movements dull into shallow spasms does the spider deliver the second stage of her attack. She positions her chelicerae, twin mouthparts tipped with sharp fangs, at the grasshopper's exoskeleton. The bite is small, almost imperceptible, but its effects will spread far beyond the entrance wound. The venom she injects is a potent mixture of enzymes and neurotoxic molecules, each one crafted by evolution to dismantle an insect body from the inside outward. The first wave consists of membrane-disrupting compounds. These chemicals pry apart the protective layers around individual cells, allowing fluids to leak and enzymes to flow where they should not. The grasshopper's tissues begin to lose integrity, starting at the bite site and radiating outward like ink spreading in water. Within the hemolymph, the insect equivalent of blood, other venom components start breaking down proteins and complex carbohydrates. Digestive enzymes that normally operate in the spider's gut now travel through the prey's body, liquefying organs into a nutrient-rich soup the spider can later suck out. This is not an instantaneous kill. In nature, speed is not always the goal. Efficiency is. The wasp spider's method ensures that the grasshopper's internal structures are converted into consumable fluid without wasting energy on unnecessary destruction. The silk immobilizes and suffocates. The venom disables and digests. It is a double attack. Two biological systems evolve to work in seamless partnership. As the venom circulates, the camera moves inside the grasshopper's body with imagined microscopic perspective. Hemocytes, the insect's immune cells, rush toward the wound in an attempt to contain the invading toxins, but they are overwhelmed. Venom components specifically target these defensive cells, rupturing them before they can mount an effective response. Muscles soften, neural pathways misfire, and the once powerful limbs that drove impressive leaps in open grassland fall into irreversible stillness. Outside, the spider checks the cocoon, tapping it with precise leg movements that transmit vibrations through the silk. These taps are a way of assessing the internal state of the prey. Certain vibration patterns indicate stillness, softness, and readiness for consumption. Once the vibrations reveal the correct combination of silence and texture, she begins the feeding process, piercing the wrapping silk to reach the liquefied tissues beneath. There is no ripping or tearing involved. The meal is already pre-digested by the venom. The efficiency of this system becomes even clearer when the broader ecological context unfolds. Grasshoppers are herbivores that can rapidly deplete vegetation in warm seasons. By targeting them, wasp spiders play a role in regulating plant-eating insect populations. Their webs, placed strategically in medium-height vegetation, intercept species that move between ground-level feeding sites and resting perches. The stable omentum, the zigzag silk structure, may further attract prey by reflecting ultraviolet light in patterns that mimic gaps in foliage, visual illusions detectable to many insects. The wasp spider herself is adapted for this ambush lifestyle. Her large abdomen stores silk-producing proteins. Her vision detects both motion and contrast, and her legs carry special claws for manipulating strands with near-surgical precision. Unlike wasps that rely on venom to subdue prey before dragging it back to a nest, the wasp spider relies equally on engineering and chemistry. She does not chase. She waits, anchored in a web held taut like a silent trap, and lets physics and instinct do the rest. The chapter closes with a wide shot of the meadow once more. The grasses continue swaying gently, hiding 1,000 webs between their stems. For every one insect that escapes into open air, another meets an end inside a silk cocoon, where oxygen dwindles and venom begins its quiet command. In this world, survival does not depend solely on speed or strength, but on the ability to master both structure and chemistry. A combination the wasp spider executes with unerring precision. At the edge of a warm forest clearing, where sunlight pours through the canopy like a slow-moving river of molten metal, a glimmer catches the eye long before the spider itself becomes visible. Suspended between two broad leaves is a web unlike any other in the surrounding vegetation. Its silk does not merely reflect light, it gathers it, bending each ray into threads of soft gold that glow even when the sun shifts behind drifting clouds. This is the architecture of the golden silk orb-weaver, a spider whose craftsmanship has been refined over millions of years until it surpassed the purely functional and stepped into something almost otherworldly. The camera moves closer, and the gold intensifies. At high magnification, each strand thickens into a tube of translucent amber, shimmering as it absorbs and refracts the sunlight. Orb-weaver silk contains unique pigments and structural proteins that give it this rare coloration, a hue that scientists believe may serve multiple ecological purposes. It may lure insects that are drawn to bright patches of light, or it may mask the web's presence by blending with the sunlit background. Experimental studies suggest the silk may even interfere with certain wavelengths visible to insects, blurring their perception just long enough for the web to catch them unprepared. But whatever its precise evolutionary advantages, one thing is certain: the golden web stands out as one of nature's most beautiful traps. The spider waits at its center, legs splayed with measured symmetry, each one resting lightly upon a radial line. These threads function as sensory conduits, carrying information about air currents, wing beats, and the frantic vibrations of struggling prey. When an insect collides with the web, the entire structure responds like a tensioned musical instrument. Even a faint touch can send ripples through dozens of connected strands, allowing the orb-weaver to pinpoint the exact position of the disturbance within moments. A moth, drifting lazily through the clearing, makes that fatal mistake. Drawn perhaps by the reflective sheen of the silk, or confused by the way it blends into the backlit leaves, the moth sails directly into the web. Its wings slap against the sticky spiral fibers. These particular threads, coated with adhesive droplets, stretch and contract like elastic, distributing the shock of impact while ensuring the moth cannot break free. The more the moth beats its wings, the deeper it sinks into the sticky loops. The orb-weaver reacts instantly. She darts along the radial lines with surprising agility, guided by shifts in tension. As she approaches the moth, the camera zooms in on the fine sensory hairs that cover her legs. These hairs, called trichobothria, detect minute vibrations in the silk. They help the spider interpret not only the location of the prey, but also its size, strength, and level of threat. A large insect struggling violently can damage the web. A venomous intruder, like certain bees or wasps, can injure the spider. But a moth is neither large nor dangerous. It is simply trapped, its struggles already slowing as fatigue begins to outweigh desperation. Before the spider deploys her silk wrapping technique, she must ensure the prey cannot fight back. While the bite of the golden orb-weaver is not designed to kill instantly, it does something equally effective. It disrupts the ability of muscles to respond to neural commands. The spider positions her chelicerae at the edge of the moth's thorax and delivers a precise venom injection. The venom diffuses quickly through the hemolymph. Inside the moth's body, neural signals, which normally travel through motor neurons and stimulate muscle fibers, begin to falter. Orb-weaver venom contains peptides that block specific ion channels in nerve cells. These channels regulate the movement of charged particles essential for transmitting electrical impulses. When these impulses fail, muscle contractions weaken and stop. In the moth, this manifests as a sudden, jerky tremor, followed by a wave of paralysis creeping across the wings and legs. The insect continues to twitch for a few moments, but the movements become increasingly uncoordinated, like signals cut off mid-transmission. Finally, the wings stiffen, then fall slack. Only now does the spider begin the second phase. Using her hind legs and spinnerets, she releases a different type of silk, strong, dry, and non-sticky, to wrap the prey. This wrapping silk is designed for speed. The spider moves in tight arcs around the moth's body, spinning band after band until the insect's wings are pinned against its thorax. The paralysis induced by the venom ensures that the wrapping process is efficient and safe. If the moth were still capable of powerful wing beats, it could tear the web apart. But with its nerves compromised, it can only shudder weakly as layer after layer of silk seals it into a compact bundle. The golden silk orb-weaver's strategy reflects a sophisticated balance between web mechanics and venom chemistry. The web serves as the first line of capture, draining the prey of energy through struggle. The venom serves as the second, shutting down motor function so the spider can secure the prey without risking damage to her fragile architecture. Together, they form a two-step system that maximizes efficiency and minimizes danger. As the spider completes her wrapping, a second insect collides with the web. This time it is a small fly, drawn into the sunlit clearing where the golden strands mimic the reflections of dew-covered leaves. Unlike the moth, the fly hits the spiral threads at an angle and immediately begins a frantic series of flips, trying to use its legs to pry the sticky fibers off. For a moment it appears the fly might escape, but the more it rotates and pulls, the more silk adheres to its limbs, and the stickier the situation becomes. The orb-weaver moves again, delivering a rapid bite before the fly's momentum threatens to tear the delicate geometry of the web. The venom sinks into the fly's body, and its spasms shift into sharp, stiff pulses. These spasms are not signs of resistance, but the physiological effects of nerve disruption. The toxins interfere with synaptic transmission, causing muscles to contract uncontrollably before collapsing into full paralysis. Once again, the prey becomes limp, offering no meaningful resistance to the wrapping process. The golden coloration of the web plays a subtler role in these encounters. Recent studies suggest that the web's brilliance may influence how insects perceive the environment. Some species are attracted to ultraviolet reflections. Others are confused by bright, continuous patterns that resemble gaps between leaves or openings in the canopy. Regardless of the mechanism, the golden silk orb-weaver gains an advantage. A well-lit web is more visible to predators like birds, but the spider accepts that risk because the same visibility increases her chances of capturing prey. Meanwhile, the warmth absorbed by the web can dry dew faster and maintain structural integrity in cooler morning hours, ensuring the web remains functional through shifting temperatures. As the day continues, the orb-weaver maintains her position in the web's central hub. She adjusts tension lines, devours damaged threads, and spins new ones to repair gaps left by the morning's captures. Every part of her behavior reflects energy optimization. Spinning silk is metabolically expensive, so she recycles proteins whenever possible. Venom is precious, so she injects only the amount necessary to paralyze prey, rather than kill it outright. By evening, the light softens, and the golden web shifts from bright amber to muted bronze. The prey wrapped earlier remains suspended, preserved by venom-induced paralysis and silk confinement. Later, the spider will feed by injecting digestive enzymes and drinking the liquefied tissues. But for now, she simply waits, poised at the center of a masterpiece that functions as both trap and extension of her senses. In this ecosystem, survival is not about brute force. It is about the ability to manipulate the laws of physics, chemistry, and perception. The golden silk orb-weaver does this with threads spun from her own body and toxins refined across evolutionary time. Each strand, each bite, and each captured insect tells the same story. A world crafted with patience, precision, and a golden shimmer that hides the silent collapse of nerves within. Night settles over the desert with a swiftness that feels almost mechanical, as if the sun has been switched off rather than allowed to slip beneath the horizon. Temperatures drop in long, smooth gradients, releasing the heat that had been trapped in sand and stone throughout the day. Under this cooling canopy, creatures that hid from the punishing sun begin to stir. The desert at night is not silent, it simply shifts to a different frequency. Where daytime offers the chirring of birds and the scurry of lizards, nighttime pulses with slower, heavier rhythms. The footsteps of nocturnal hunters, the wing beats of moths, and the quiet scrape of something armored moving like a shadow across the soil. A black scorpion emerges from beneath a rock outcrop, its body casting a faint reflective sheen beneath the moonlight. To the naked eye, its form is nearly invisible, blending into the dark substrate. But under ultraviolet illumination, its exoskeleton glows with an otherworldly blue-green radiance. This fluorescence, caused by compounds embedded in the scorpion's cuticle, has puzzled biologists for decades. Some propose that it helps the scorpion detect light levels or navigate the landscape. Others suggest a role in communication or camouflage. Whatever the reason, the effect is mesmerizing. A ghostly outline drifting across the sand like a living constellation fallen from the sky. The camera settles into slow motion as the scorpion lifts its tail. The arch is deliberate, rising from the abdomen in a smooth curve until the stinger hovers just above the creature's head. The posture is iconic, a silhouette recognized even by those who have never encountered a scorpion in the wild. But beneath this intimidating posture lies a remarkably nuanced system of control. The scorpion is not simply preparing to strike, it is calibrating, deciding how much venom, if any, to deliver. Unlike many venomous animals that release the same chemical mixture with each sting, scorpions possess an extraordinary ability to modulate their venom output. This behavioral control functions like a living dial. A warning sting may be delivered without venom at all, a so-called dry sting designed to deter larger threats without wasting precious biochemistry. Venom is metabolically expensive to produce, requiring energy and specific nutrients that are not always abundant in harsh ecosystems. For scorpions, conservation is essential. Thus, the decision to inject venom is precise, strategic, and often reserved for moments when the benefits clearly outweigh the cost. Tonight, the target is not a large predator, but a small vertebrate, perhaps a juvenile mouse drawn to the scent of seeds scattered by wind. The mouse pauses near a tuft of grass, whiskers quivering as it surveys the terrain. The scorpion detects the vibrations of the mouse's footsteps long before it sees the creature itself. Sensory hairs along its legs pick up subtle tremors, spreading through the ground, converting mechanical waves into neural signals. This vibration sensitivity is so refined that the scorpion can determine the direction, distance, and even the approximate size of approaching animals with astonishing accuracy. The strike, when it comes, is a burst of movement so fast it appears almost instantaneous. The tail whips downward with a velocity that belies the scorpion's otherwise slow demeanor. The stinger, shaped like a curved blade tipped with a sharp point, punctures the mouse's skin in a clean, singular motion. The scorpion delivers a controlled dose, not a warning, but not the full arsenal either. Instead, it releases enough venom to immobilize quickly and ensure the prey cannot escape. Inside the mouse's body, the venom begins its work. Scorpion venom is not a single toxin, but a complex mixture of peptides, proteins, and salts, each designed to target specific components of the nervous system. Many of these molecules interact with ion channels embedded in nerve cell membranes. Ion channels regulate the flow of charged particles, such as sodium and potassium, across the membrane, and this flow generates the electrical impulses that allow nerves to communicate with muscles. When venom peptides wedge themselves into these channels, they hold them open or force them shut. In either case, the electrical system breaks down. Pain becomes the first symptom. Specialized venom components activate sensory neurons that respond to tissue damage, sending signals of burning, stinging discomfort through the nervous system. In small animals, this pain rapidly escalates into disorientation as the nervous system starts to misfire. The mouse's muscles twitch in quick, uncontrolled spasms. The venom affects not only motor neurons but also the muscles of respiration. If enough venom is delivered, the diaphragm, the primary muscle used for breathing, becomes paralyzed, leading to respiratory collapse. In many cases, this happens in mere moments, leaving the prey motionless before it fully understands what has struck it. With the mouse incapacitated, the scorpion advances slowly, using its pedipalps, large claw-like appendages, to grasp the prey. These pincers are strong enough to hold struggling animals, but are often secondary when dealing with small, soft-bodied creatures. The venom has already completed most of the work. As the scorpion begins feeding, enzymes in the venom help break down tissues, making the meal easier to consume through the scorpion's narrow oral opening. Here, venom serves not only as a hunting tool, but also as a digestive aid. Insects, too, fall victim to the black scorpion's dual-purpose weaponry. A beetle wandering across the sand experiences the same sequence: a rapid strike, a calculated dose, and a near-instant shutdown of muscular coordination. The beetle's exoskeleton may offer some protection from crushing forces, but it cannot shield the internal nervous system from venom molecules that slip past barriers and attach themselves to ion channels with surgical precision. In the broader ecosystem, the scorpion's venom strategy plays a role far beyond its own survival. By regulating populations of rodents, insects, and other small animals, scorpions help maintain the delicate balance of desert environments. Too many herbivorous insects could strip vegetation and destabilize soil. Too many rodents could alter seed dispersal patterns. The scorpion's predation helps hold these forces in check, contributing to an ecological equilibrium maintained through countless subtle interactions. Interestingly, scorpions do not always rely on venom when hunting. If prey is small enough to subdue with physical strength alone, the scorpion may opt for mechanical capture, conserving its chemical reserves. But when faced with threats or prey capable of escape or counterattack, the venom dial turns upward. The scorpion can release a rapid-acting mixture designed for pain alone, a deterrent aimed at larger animals, or it can deliver a paralytic dose intended to immobilize. In some species, venom composition can even shift between defensive and offensive uses, demonstrating an evolutionary flexibility that few predators possess. The camera returns to ultraviolet illumination as the scorpion retreats into a crevice after feeding. Under this ghostly glow, its entire body radiates color against the dark stone. Scientists still debate whether this fluorescence acts as a form of photoprotection, warning, or sensory enhancement. Whatever its purpose, it creates an uncanny image, a creature built for the night, glowing silently in a wavelength invisible to most eyes, but captured here in vivid, spectral detail. In the desert landscape where survival hinges on precision, efficiency, and the ability to strike decisively, the black scorpion stands as a master of chemical control. Its venom is not a blunt weapon, but a finely tuned instrument capable of delivering pain, paralysis, or warning, depending on the moment's needs. With every sting, the scorpion balances metabolic cost, ecological opportunity, and evolutionary strategy. A predator guided by both instinct and biochemical sophistication. At dawn, when the first rays of sunlight thread between leaves and lay narrow beams across the forest floor, a creature no larger than a grain of rice rises into alertness. Dew still beads on the moss, tiny lenses refracting the morning light into miniature rainbows. The air is cool, carrying the faint tremor of wings from insects beginning their daily foraging flights. In this soft, filtered glow, a jumping spider positions itself on the edge of a sunlit twig, its body still, its eyes impossibly focused. Those eyes, 8 in total, define its world. The 2 central front eyes, enormous relative to the spider's size, resemble polished black domes. They grant the spider a depth perception that rivals that of animals hundreds of times larger. Each eye contains layers of retinas capable of shifting focus in ways that allow the spider to judge distance with exquisite precision. The camera moves closer, revealing subtle shifts in the reflective surfaces, microscopic adjustments that align vision with the flutter of a fly passing overhead. The fly lands on a curled leaf just a few centimeters away. It cleans its forelegs, unaware of the predator measuring every movement. To human senses, this gap seems trivial, but for a spider whose entire body measures only a few millimeters, it is a vast chasm. Yet the jumping spider does not consider it an obstacle. It calculates trajectory, wind currents, and landing stability with a brain no wider than a pin's head, compressing complex geometry into instinctive action. The spider crouches, its abdomen dips. Internal hydraulic pressure builds within its legs. Jumping spiders do not leap using muscle contraction alone. Instead, they force hemolymph, the spider equivalent of blood, into their leg joints, increasing pressure until the legs extend explosively. This hydraulic mechanism behaves like a biological spring, storing potential energy and releasing it in an instant. In slow motion, the leap unfolds frame by frame. The spider launches into the air with astonishing acceleration, covering a distance more than many times its body length in less than the blink of an eye. Its trajectory is perfectly aligned with the fly's dorsal thorax. Even as the spider arcs forward, it releases a silk safety line behind it, a lifeline that ensures survival should the attack fail. This defensive silk, barely visible even under magnification, anchors the spider to its starting point. The fly begins to turn, its wings flicking as it senses air disturbance, but the reaction is too late. The spider lands squarely on its target, legs locking around the fly's thorax with delicate yet unyielding precision. Before the fly can beat its wings, before a single coordinated muscle contraction can lift it from the leaf, the spider executes the heart of its strategy, a bite delivered with surgical minimalism. Jumping spiders possess small, needle-like fangs positioned just beneath their eyes. These fangs house micro-reservoirs of venom, a liquid measured in volumes so tiny that human instruments struggle to quantify them. Yet these droplets, infinitesimal in size, carry biochemical power perfectly calibrated for the spider's needs. The fangs penetrate the soft membrane between segments of the fly's exoskeleton. A weak point the spider locates with intuitive accuracy. The venom enters with astonishing speed. It is not a brute force toxin designed to cause widespread tissue destruction. Instead, it targets synapses, the tiny junctions where nerves communicate with muscles. Many jumping spider venoms contain peptides that block neurotransmitter release or interfere with receptor binding. These micromolecules effectively shut down communication between nerve cells and the muscles they control. The result is paralysis, not by collapse, but by silence. The nervous system simply stops sending messages. The camera shifts to a microscopic view, illustrating the venom's path. Neurons attempt to fire, sending waves of electrical signals down their lengths. But the venom has already wedged itself into the machinery that releases neurotransmitters. Synaptic vesicles fail to open. Ion channels remain stubbornly closed. Without chemical messages, the muscles cannot respond. The fly's wings twitch once, twice, then fall still. What makes this biological strategy extraordinary is not the potency of the venom itself, but the precision with which it is used. Jumping spiders do not inject unnecessary amounts. They deliver exactly what is required to immobilize prey of a particular size and nervous complexity. This makes the venom extraordinarily efficient and metabolically economical. Some researchers describe this as sniper venom, toxin applied with near-perfect aim, minimal volume, and maximum effect. Experiments conducted in laboratory settings reveal just how fast this paralysis occurs. When flies are exposed to controlled microdoses of jumping spider venom, their reaction times, normally measured in tiny fractions of a second, vanish almost immediately. Movements that would normally be reflexive, such as wing flicks or leg withdrawals, cease within moments. A fly that moments earlier could detect and evade the swipe of a human hand becomes incapable of coordinating even the simplest motion. The spider holds the fly tightly as the paralysis takes hold. But unlike many predators that immediately begin feeding, the jumping spider often waits for the venom to complete its work. Its eyes remain focused, scanning for threats or rival spiders that may attempt to steal the kill. When safety is assured, it begins feeding by injecting digestive enzymes that liquefy soft tissues. The spider then drinks the nutrient-rich fluid, leaving behind the hollow exoskeleton. This hunting strategy reveals a broader ecological truth. Jumping spiders are not mere ambush predators. They are active visual hunters. Their reliance on precision over force allows them to pursue prey larger or more agile than themselves. This adaptability contributes to their success across ecosystems ranging from tropical forests to temperate grasslands and even urban gardens. The scene widens to show the surrounding habitat. Sunlight intensifies, warming the moss and driving moisture into the air. Small insects buzz through shafts of light like drifting sparks. The jumping spider now moves on, its hunt complete. It scans its environment with minute head tilts, adjusting visual depth and refocusing each eye independently. This combination of high-resolution vision and microdosed venom elevates the spider far above most other small predators. The spider soon encounters another insect, a leafhopper, resting in a patch of shade. The leafhopper's powerful hind legs can propel it into sudden leaps that leave predators flailing in empty space. But the jumping spider does not chase blindly. It circles slowly, using foliage to break its silhouette. When it leaps again, the same choreography repeats. Hydraulic launch, perfect alignment, tiny puncture, instantaneous collapse of coordination. The leafhopper's muscles stiffen mid-motion, its escape potential erased by an almost invisible dose of venom. As the day progresses, the spider shifts between hunting, grooming its sensory hairs, and moving to new vantage points. Every movement is precise, every leap is calculated, every bite is delivered with exacting restraint. The chapter settles with the spider perched atop a dew-kissed stem, its eyes catching the last glints of sunlight. It has no interest in waste, no need for excess. Evolution has sculpted a predator whose power lies not in size or brute strength, but in the ability to deliver microscopic chemistry with pinpoint accuracy. A hunter whose weaponry is measured not in volume, but in perfection. In a world where survival often hinges on overwhelming force or defensive armor, the jumping spider represents an alternate path. Mastery through precision. Its venom may be small in quantity, but its effect on the miniature nervous systems it targets is absolute. A single bite, delivered with quiet confidence, is enough to freeze a life mid-motion. The rainforest dawn begins, not with birdsong, but with the slow awakening of heat. Moisture clings to every leaf, gathering into droplets that shimmer as diffused sunlight threads through the canopy. Mist lifts from the forest floor in pale ribbons, curling between roots and decaying trunks. Beneath this quiet surface layer, the first tremors of movement begin, soft vibrations running through soil rich with fungal threads and ancient nutrients. These vibrations do not come from something large and lumbering. They come from countless tiny feet moving in perfect synchrony. A column of leafcutter ants emerges from the shadowed undergrowth, their bodies glistening with humidity. Workers march in tight formation, their segmented antennae sweeping the air like biological radar. Unlike many ant species that forage singly, leafcutters operate as a unified industrial force. Their strength lies not in individual size, for each worker could fit easily atop a fingertip, but in collective precision, a seamless choreography honed by millions of years of evolutionary pressure. At first glance, their mission appears routine, harvesting leaves for their subterranean fungus gardens. But today, the scene shifts. A caterpillar, thick-bodied and luxuriously soft, has wandered into the ant trail. It lies across the path, sluggish from the morning cool, an unintentional blockade. In most forests, a caterpillar this size would be insignificant. But here, it is a spark dropped into a powder trail. The first ants encounter it head-on. They pause, antennae brushing across its fleshy surface. The caterpillar lifts its head, slow and uncertain, sensing the disturbance. But by the time it registers danger, the workers have already sent chemical messages back through the column. Alarm pheromones disperse in rapid waves, summoning reinforcements with the immediacy of a silent siren. From the surrounding vegetation, soldiers descend. Larger, broader-headed ants whose mandibles open like sharpened shears. The air vibrates faintly from the rush of coordinated bodies converging on the intruder. The caterpillar, now recognizing the threat, begins to thrash, but movement only worsens its situation. Every twitch attracts more mandibles, more workers, more chemical readiness. The first bite lands near the caterpillar's flank. Leafcutter ants do not rely on brute crushing force alone. Their mandibles, lined with microscopic ridges, clamp down with a slicing efficiency that belies their size. The caterpillar's soft tissue yields instantly, but the mandibles are only the anchoring stage. What follows is the true weapon. Venom sacs within the ants' bodies begin releasing formic acid. This acid is not a mild irritant. It is a potent chemical capable of burning through delicate membranes and disrupting cellular structures. Workers position themselves strategically, bracing their legs as they compress their abdomens to eject the acid through small openings near the mandibles. Each injection is tiny, but multiplied across dozens, then hundreds, the effect is catastrophic. The caterpillar's skin blisters where acid droplets land. Under magnification, the damage is immediate. Formic acid dissolves lipids in cell membranes, allowing internal fluids to leak outward. Tissue softens, darkens, collapses. The caterpillar writhes, but its movements have already become uncoordinated. The ants have targeted joints, spiracles, and vulnerable seams where soft tissue lies, close to the exterior. More ants climb its sides. Some latch onto hairs or folds of skin, using these footholds to secure their positions. Others swarm beneath it, biting into the soft underside. From every angle comes another burn, another chemical sting. The assault is not a single decisive strike, but a relentless accumulation of micro-attacks, 1,000 tiny chemical burns delivered with tireless precision. Leafcutter ants are often celebrated for their agricultural abilities, cultivating fungus in vast subterranean chambers. But beneath that constructive role lies an army fully capable of overwhelming creatures many times their size. Their coordination results from a mix of pheromone trails, tactile communication, and instinctive pattern recognition. When one ant injects formic acid into a wound, the smell of the chemical itself becomes a cue for others to target the same area. Chemical warfare, in this sense, is not merely deployed, but amplified through group behavior. As the caterpillar's struggles weaken, soldiers take over the most dangerous roles. Their larger mandibles can cut deeper and hold tighter. A soldier clamps firmly onto one end of the caterpillar, while several workers begin slicing through its opposite side. This creates leverage, exposing more tissue for acid application. The air fills with the sharp, acrid scent of formic acid, a chemical signature of the ant's escalating attack. The caterpillar's breathing shifts into frantic pulses. Insects breathe through spiracles, and the ants know this instinctively. They coat these openings with acid, burning the delicate membranes inside and restricting airflow. The caterpillar begins suffocating even as its tissues degrade. The dual pressure of tissue destruction and respiratory collapse creates a downward spiral that few intruders survive. One might assume that leafcutter ants use formic acid solely as a weapon, but the chemical has multiple roles in the colony. It disinfects surfaces, sterilizes wounds, and protects the fungal gardens from harmful microbes. In battles, these secondary benefits become primary advantages. every droplet of acid the ants release. Not only injures prey, but also prevents opportunistic bacteria from colonizing the carcass before the ants can process it. As the caterpillar finally lies still, the assault does not stop. Ants continue applying acid, ensuring that no bacterial bloom interferes with the upcoming harvest. Soldiers reposition themselves to hold the body steady, while workers begin cutting the softened tissue into manageable fragments. Each piece will be carried back to the nest, not as food for adult ants, but as substrate for their fungus, a cycle of transformation where chemical violence becomes agricultural fuel. Occasionally, the ants must defend their kill from opportunistic predators, parasitic flies, small lizards, or rival ants drawn by the scent of exposed tissue. In these moments, leafcutter colonies reveal another advantage: sheer numbers. Reinforcements stream in along the pheromone trail, forming living walls around the carcass. New soldiers take positions with mandibles raised. The acid reservoir is far from empty. The camera shifts outward, capturing the landscape around the scene. The forest floor is alive with decomposers, herbivores, and predators, but few species command the environment as thoroughly as leafcutters. Their trails form highways, their pheromones direct traffic, their nests reshape soil structure, and when necessary, they unleash coordinated chemical warfare capable of erasing animals many times their mass. To understand their evolutionary success, one must see the system as a whole. Workers are the gatherers, soldiers are the enforcers, and acid is both weapon and tool. No single ant could accomplish what the colony achieves collectively, but together they create a living engine, one that cuts, burns, dissolves, and transports, never hesitating, never disputing direction. The chemical efficiency is chilling. 1,000 tiny burns, each insignificant alone, combine into a force larger than the sum of its parts. Now the caterpillar is gone. Only fragments remain, carried piece by piece into the dark tunnels where fungus will transform animal tissue into nourishment for the colony. The ants disperse, the trails slowly dissolving back into the forest's ambient movement. The undergrowth, for a moment disturbed, returns to stillness. But the message lingers in the air like the fading scent of acid. In the rainforest, power is not always measured in size or singular strength. Sometimes it lies in numbers, coordination, and the ability to wield chemistry as both blade and fire. The leafcutter army does not rely on one kill shot. It brings 1,000. In a meadow washed with late morning light, where grasses sway gently and pollen drifts in golden clouds, a small red shape dots the green expanse like a misplaced seed of paint. At first glance, the ladybug seems almost ornamental, a tiny rounded jewel perched on a blade of grass. Its shell gleams with hues that feel almost festive, bright crimson punctuated with deep black spots. To human eyes it reads as harmless, cheerful, a welcoming symbol of gardens and childhood curiosity. But in this ecosystem, where every color and scent carries a survival message, the ladybug's appearance is not mere decoration. It is a brand, a signature, a biological warning that most animals learn quickly and never forget. The ladybug crawls with deliberate calm, its antennae tapping the leaf surface as it searches for aphids hidden in clusters along stems and shoots. Its feet grip with fine precision, and its domed elytra, the iconic hard wing covers, catch sunlight in a way that enhances its bright red saturation. Predators scanning the meadow from above may see it easily, but that visibility is part of its strategy. The red stands out because it must. It is a promise of consequence. On a nearby branch, a small bird watches the beetle's slow progress. A fledgling still refining its hunting technique, the bird is driven more by instinct than experience. Hunger presses it forward, and the bright red beetle, plump and slow-moving, appears to be an excellent target. The bird hops closer, head tilting as it evaluates distance. Then, in one smooth motion, it strikes, grabbing the ladybug between its beak tips. For a heartbeat, the scene holds still. The ladybug is trapped, yet it does not thrash or fight. Instead, something begins to happen at its joints. at the seams where its legs meet its body. From these tiny fissures, droplets of bright yellow fluid begin to well up, then spill outward. The fluid is thick, glossy, and impossibly vivid against the red shell. This is reflex bleeding, a defense mechanism evolved over millions of years, and the liquid now coating the beetle's legs is laced with bitter alkaloids that advertise danger in the most intimate way possible, through taste. The bird's beak barely touches the fluid before the chemical message reaches its senses. Alkaloids in the ladybug's blood attack receptors that detect bitterness and toxicity, triggering an instinctive recoil. The bird jerks backward, shaking its head violently. The beetle drops to the ground, tumbling through a few blades of grass before landing upright, an evolutionary bonus of its rounded shape. The bird, meanwhile, is still gagging, its beak wiping against branches and leaves as it tries to rid itself of the taste. The scene is not simply anecdotal. It mirrors the results of structured experiments conducted by researchers who study predator-prey learning. When naive predators, birds, lizards, or even small mammals, encounter a ladybug for the first time, they often attempt to eat it. But after tasting the bitter, toxic hemolymph, they develop long-lasting aversions. In controlled settings, many predators refuse to attack anything with the same red and black pattern for weeks, sometimes months. The visual memory becomes deeply imprinted. Red shell with black dots equals chemical regret. Back in the meadow, the fallen ladybug rights itself and moves again, leaving behind small yellow smears on the leaf. Reflex bleeding is costly. It requires the beetle to sacrifice fluids rich in nutrients and defensive compounds. But the reward is survival, and the beetle's slow, composed gait resumes as though nothing occurred. The bird hops farther away, fussing at its beak and shaking its head. This single encounter has reshaped its future behaviors. The ladybug's warning colors, known as aposematic coloration, are not arbitrary. They serve as honest advertising for its chemical defenses. Unlike animals that rely on camouflage or speed, the ladybug relies on being conspicuous. By broadcasting its toxicity, it avoids unnecessary conflicts, conserving energy that would otherwise be spent on physical escape. The camera shifts to ground level. The ladybug investigates a dense cluster of aphids feeding on a stem. These soft-bodied insects are its primary prey, and as it moves, its mandibles begin to work, snipping aphids one by one. This predatory role gives ladybugs immense ecological value in gardens and natural habitats, reducing pest populations that might otherwise devastate plant communities. Yet predators do not see the insect as a gardener's helper. They see a potential meal, until they learn otherwise. Not all predators are naive. Some spiders, particularly orb-weavers, cannot rely solely on visual recognition. Their trap lines detect movement, not color. When a ladybug becomes ensnared in a web, the spider approaches cautiously, tapping threads with its front legs to evaluate the prey. The ladybug, realizing the threat, curls its legs inward and begins to exude its characteristic yellow fluid. Even spiders, whose taste receptors differ greatly from those of birds, react strongly to the alkaloids. Many release the beetle after the first contact, allowing it to fall from the web and escape. A few highly specialized spiders may attempt to wrap and isolate the chemical source, but the majority quickly learn that handling a ladybug leads to an unpleasant chemical encounter. The alkaloids in question, often derivatives of compounds known as coccinellines, function by interfering with cellular communication in predators. They disrupt certain metabolic pathways, triggering nausea or neural discomfort. For small vertebrates, even a tiny exposure can cause temporary vomiting or difficulty swallowing. Although the venom is not designed to kill, its effectiveness lies in triggering memory. In nature, a predator who remembers what not to eat wastes less energy and avoids unnecessary suffering. This dynamic reinforces the ladybug's aposematic advantage over generations. The camera offers a closer view of the beetle's leg joints, where the bright yellow fluid continues to bead. These joints, normally hidden beneath the glossy exterior, become flag-like portals for chemical release. Ladybugs possess specialized reservoirs for their toxic hemolymph. When threatened, muscles contract to force the fluid outward. The process is instantaneous, offering no delay between attack and chemical defense. Remarkably, The beetle's body is designed to withstand this self-induced bleeding without losing critical fluid volume. Reflex bleeding is both a last resort and a calibrated tool of survival. As the day warms further, more predators move through the environment. A praying mantis, perched high on a reed, swivels its triangular head with uncanny precision. Its eyes fix on the ladybug below. The mantis, however, is not deterred by visual warnings alone. Its hunting instinct outweighs caution. In a sudden movement, it lunges and grabs the beetle with its spined forelegs. The ladybug's defense triggers again. Yellow droplets coat the mantis's grip. The mantis attempts to bite, but hesitation creeps in. Chemical cues reach its palate. The mandibles open, then close again in confusion. Finally, the mantis drops the ladybug. The beetle hits the leaf surface and rolls, but survives. The mantis cleans its forelegs with visible discomfort, repeatedly rubbing them against its abdomen. Experiments with mantises show similar outcomes. After 1 or 2 attempts, most individuals avoid red and black beetles altogether. Vision becomes a memory-linked filter, preventing repeated mistakes. The ladybug's color pattern, seemingly simple, has become a lifelong cue for predators. This is not food. As afternoon light deepens, the ladybug climbs higher along a stalk, its shell gleaming as though polished. It has survived not because it is fast or strong, but because it wears a uniform that speaks clearly across species. Its toxins are not lethal weapons like those of a wasp. They are pedagogical tools, teaching lessons that resonate through predator communities generation after generation. The meadow returns to quiet. The bird hunts elsewhere. The spider repairs its web. The mantis resumes scanning for less costly prey. And the ladybug, ever bright, ever deliberate, continues its journey across a world that knows better than to underestimate a cute red shell that bleeds poison. The afternoon sun settles over a patch of dry meadow with a heat that seems to thicken the air itself. The ground, cracked in places from days of warmth, hosts small islands of fallen leaves and seed husks. From a distance, these scattered patches appear motionless. But as the camera glides closer, the stillness dissolves into something startling. A sprawling crowd of insects. Dozens, then hundreds, then seemingly thousands, gathers in a tight, pulsing mass of red and black. Firebugs. Their bodies, lacquered in saturated crimson and coal dark patterns, transform the earth into a mosaic of warning colors. The cluster moves slowly, shifting like a single breathing organism, individuals climbing over one another in restless waves. Their coloration is so bold it feels almost defiant, as though each beetle is broadcasting the same message. We dare you. The firebug's red and black armor is no accident of aesthetics. It is a posmatism made blatant, a visual megaphone shouting chemical truth. Where other insects rely on camouflage or speed, firebugs choose exposure. Their colors demand attention, from mates, from rivals, and especially from predators. To birds scanning the area for an easy meal, the colors are unmistakable. Red equals risk. Black spots equal consequence. The camera zooms in on a single firebug moving across a fallen seed pod. Its legs tap rhythmically, antennae sweeping the surface as it probes for edible traces. Firebugs feed on seeds of plants like mallows and linden trees, many of which contain compounds that are far from harmless. Alkaloids, terpenoids, and other defensive molecules evolved by plants to deter herbivores form the chemical barricade within these seeds. Many insects avoid them entirely. Firebugs do not. Instead, they pierce the seed coat with their needle-like mouthparts, suck out the internal fluids, and ingest the chemical defenses as part of their nutritional intake. But what would poison other insects becomes a foundation for the firebug's own survival. Inside their digestive system, firebugs metabolize these plant toxins, breaking down some compounds while storing others. The result is a personal chemical arsenal. Bitter, foul-tasting, gut-disrupting molecules that coat their internal tissues and, when necessary, reach the outside world. This transformation of plant poison into insect armor is a remarkable example of evolutionary recycling. The plants develop toxins to protect themselves from herbivores. The herbivore evolves a way to repurpose those toxins against its own predators. And over generations, predators learn through harsh experience to avoid the brightly colored insects that have mastered the chemical relay. The camera shifts to a nearby bush, where a small bird hops along a branch. It surveys the ground with a practiced predatory intent. Its eyes catch the red and black gathering immediately. The pattern triggers a flicker of recognition, but hunger nudges caution aside. The bird swoops down, landing near the edge of the cluster. Firebugs scatter briefly, their movement like the ripple of a thrown stone across water. But their separation is brief. They regroup moments later, forming a defensive mass that visually reinforces their warning. The bird pecks at one of the individuals. The firebug's shell cracks slightly, releasing a faint but unmistakable scent, sharp, chemical, and bitter. The bird recoils, shaking its head as the taste hits, its beak opening and closing in discomfort. A small amount of the firebug's internal fluid has reached the bird's mouth, carrying alkaloids that cling stubbornly to sensory receptors. These compounds not only taste unpleasant, but can disrupt digestive processes, causing nausea in predators that consume too much. The bird hops away, ruffling its feathers in agitation. For a few moments, it attempts to wipe its beak on the ground, rubbing vigorously to remove the taste. Then it lifts off and flies towards safer prey. The firebugs return to their clustering as though nothing happened, their bright armor intact. This reaction is not mere anecdote. In controlled experiments, young birds exposed to firebug defensive fluids exhibit lasting aversion to red and black patterns. After a single exposure, many refuse to attack anything with similar coloration. Their learning is rapid, their avoidance durable. The firebug's coloration acts as a brand, one encounter, one taste, one mistake, and the message imprints deeply. Back in the meadow, the firebugs shift positions around a cracked seed husk. Their movements are communal and strangely rhythmic. Firebugs often aggregate in large groups, not only for warmth or mating opportunities, but also to amplify their visual threat. A single red and black insect is a warning. 100 clustered together is a biological billboard advertising danger. This gang effect magnifies their defense far beyond what any individual could achieve alone. The camera slides across the crowd. The patterns on their backs form geometric designs, triangles, circles, and elongated shapes arranged in symmetrical harmony. These markings are not random. They help predators identify the firebug even at a distance or from above. The combination of colors and shapes becomes an unmistakable signal recognized across species. To an inexperienced predator, the message says, "Try us if you dare." To an experienced one, it says, "You already know better." As the afternoon warmth intensifies, firebugs become more active. Some climb onto sunlit rocks to regulate their temperature. Their black patches absorb heat efficiently, aiding thermoregulation and allowing their metabolism to remain active even in cooler moments. Others move towards shaded areas or burrow beneath leaves to avoid overheating, a delicate balance managed through both behavior and physiology. Nearby, a small wolf spider approaches, drawn by the movement. Spiders cannot rely on color recognition the same way birds do, but they sense vibrations and scent. The spider steps cautiously toward the mass of insects. A single firebug wanders too close. The spider pounces, pinning the beetle with practiced precision. But as its fangs prepare to puncture, the firebug releases a droplet of fluid from its leg joints. Not as dramatic as a ladybug's reflex bleeding, but potent enough. The spider hesitates. Its mouthparts contact the fluid, and the chemical information travels instantly. The taste is wrong. A blend of bitterness and plant-derived toxins that interrupts the spider's feeding response. It retreats, wiping its mouthparts on the ground. The firebug, unharmed, returns to the cluster. The spider's reaction demonstrates another evolutionary advantage of firebug toxins. They operate across a wide range of predator types. Birds, lizards, spiders, and some mammals all show aversive responses to firebug chemistry. The compounds interact with gustatory and digestive systems, in ways that discourage predation through nausea or discomfort. They are not meant to kill. Death of a predator would teach nothing. The point is memory, pattern recognition, and long-term avoidance. Later in the day, the firebugs gather around several crushed seeds, their feeding synchronized. The camera reveals the fine details of their mouth parts as they pierce seed coatings and draw out sap rich with plant toxins. This steady intake ensures that their chemical defenses remain replenished. Unlike insects that synthesize their toxins internally, firebugs outsource the process to the plants they consume. They become mobile containers of recycled plant chemistry. As the sun dips toward the horizon and the shadows stretch long, the cluster begins to disperse. Small groups peel away to find shelter for the night, their armored backs still glowing faintly under the dusk light. The meadow seems to breathe easier as the red tide retreats, leaving only a few scattered individuals wandering among the grasses. Yet their legacy remains. Any predator that encountered them today will remember, not simply because of the bright colors, but because those colors carried a visceral lesson. In the natural world, honesty is often enforced through consequence. The firebug's gang colors are honest in the truest sense. A visual declaration backed by real chemical defense, bitter enough to shape the behavior of an entire ecosystem. In a world where size often dictates dominance, the firebug proves that survival can hinge on something subtler: a shared warning, a communal display, a smokescreen made not of vapor but of memory. In the sticky twilight of Madagascar's rainforest floor, where humidity condenses into a film over every stone and root, a faint rustling begins beneath a carpet of fallen leaves. The air is heavy with the scent of decay, sweet rot from decomposing fruit, the sharp tang of fungus breaking down cellulose, the earthy musk of soil teeming with microorganisms. In this dim microcosm, many insects tread carefully. A wrong meal, a contaminated puddle, or a fungal spore inhaled at the wrong moment can end a life swiftly. But there is one creature here that moves with steady, unhurried confidence, as though the environment were crafted specifically for it. A Madagascar hissing cockroach pushes aside a leaf, its glossy, armored body emerging like a small tank from beneath forest debris. Its exoskeleton glows with warm shades of brown and black, reflecting ambient light in broad curves. This species does not rely on speed or venom or stealth. Instead, it uses something far more radical, an internal chemistry lab that no other insect on the forest floor can match. The cockroach crawls toward a mound of rotting meat, the decomposing remains of a small lemur or tenrec, now colonized by maggots and filamentous fungus. For many insects, this pile would be a death sentence. The tissues are saturated with bacterial toxins, ammonia-rich fluids, and microbial compounds designed by evolution to kill competitors. But for the Madagascar hissing cockroach, this is simply dinner. It clambers onto the carcass, antennae waving slowly as it assesses what nutrients remain intact. The first surprise lies in its cuticle. Unlike the thin, easily pierced exoskeletons of many insects, the cockroach's cuticle is layered like composite armor. The outer layer contains waxes that repel moisture, essential when navigating decaying environments where wet surfaces can harbor lethal microbes. Beneath this lies a matrix of chitin and proteins, dense and flexible, offering protection not only from predators, but also from chemical penetration. Toxic fluids bead and run off its body rather than seeping inside. The camera zooms close enough to see tiny scratches and pits on the cuticle surface, each formed as the cockroach has scraped against sharp fungal hyphae or splashed through acidic pockets of decay. These scars do not threaten it. They are the marks of a creature built to endure chemical chaos. But armor alone cannot explain its resilience. The true marvel lies inside. As the cockroach feeds, powerful enzymes flow through its digestive tract. These enzymes are part of an evolutionary toolkit, allowing it to break down substances that would cripple or kill more sensitive species. Rotting meat contains toxins produced by bacteria such as Clostridium and Pseudomonas. These compounds cause paralysis, tissue destruction, and immune collapse in many organisms. The Madagascar hissing cockroach, however, possesses gut microbes and specialized detoxification pathways that neutralize these threats. Its gut microbiome functions like an internal shield wall. Bacteria living within its digestive system produce their own chemical arsenal, enzymes that degrade fungal toxins, proteins that bind and neutralize bacterial poisons, and metabolic pathways that convert harmful substances into energy sources. This partnership between insect and microbe is symbiotic in the purest sense. Without these microbes, the cockroach would be ordinary. With them, it becomes a walking bioreactor. The camera transitions to a microscopic representation. As the cockroach consumes the flesh, molecules move through its gut like swirling currents. Detoxification enzymes, such as cytochrome systems, transform harmful organic compounds into inert metabolites. Chitin-digesting enzymes help break down fungal cell walls. Even ammonia, present in dangerously high concentrations in decomposing tissues, is processed through internal filtration mechanisms that protect the insect's physiology from overload. What the hissing cockroach achieves through chemistry is equivalent to a human walking through a hazardous waste site without protective gear and leaving unharmed. Around the carcass, other insects test their luck. A beetle approaches the edge of the rotting tissue, but retreats quickly as it detects chemical cues that signal microbial danger. Fruit flies land only on peripheral areas where decomposition is minimal, but the cockroach, unfazed, continues feeding deep within the most toxic pockets. Suddenly, a vibration ripples across the forest floor. A predatory reptile, perhaps a skink or small gecko, approaches. The cockroach freezes briefly, then emits one of its signature defenses, a hiss. Unlike many insects that rely on chemical sprays or stingers, the Madagascar hissing cockroach produces sound mechanically, using spiracles, the openings along its abdomen used for breathing. By forcing air through these spiracles, the insect creates a sharp, forceful hiss reminiscent of escaping steam. In the dim understory, this sound resonates with surprising volume, startling predators unaccustomed to such noise from a small creature. Hissing serves multiple purposes. Against predators, it mimics the defensive sounds of larger animals, such as snakes. Within social groups, it acts as communication during mating or dominance disputes, and when combined with the cockroach's odor-based signals, it forms a layered defensive suite. Odor is the cockroach's second line of warning. When disturbed, it releases chemical cues detectable to other cockroaches. These signals can alert nearby individuals to danger, coordinating evasive behavior. While not toxic like formic acid or ladybug alkaloids, these odors carry information that can influence group survival. They are subtle but effective, a kind of invisible language shaped by environmental necessity. The reptile pauses, head tilted, evaluating whether this hissing, armored insect is worth the trouble. Many predators learn quickly that cockroaches offer little reward for the effort. Their bodies are tough, their defenses unpleasant, their taste often foul due to internal toxins absorbed from decaying matter. The reptile withdraws, leaving the cockroach unharmed. As night deepens, more cockroaches emerge from the forest floor. They approach the carcass from multiple angles, forming a slow-moving tide of glossy bodies. Their footsteps create soft clicks against leaf litter. Moonlight filters through the leaves, reflecting faintly off their segmented backs. This congregation is not chaotic. It is opportunity-driven, each individual responding to scent cues that lead them to nutrient-rich food sources. While they feed, they also break down the carcass, contributing to the rainforest's decomposition cycle. Without organisms like these, dead matter would accumulate, nutrients would stagnate, and the forest floor would smother under layers of decay. The cockroaches play a crucial ecological role, toxic garbage processors converting death into life-sustaining material for plants and soil organisms. Despite their reputation, these insects are not vectors of disease in the wild. Their internal systems contain defenses against harmful microbes without spreading them. Their detox pathways neutralize pathogens that could otherwise proliferate. In a rainforest defined by cycles of life and decay, the Madagascar hissing cockroach stands as a necessary intermediary. By dawn, the carcass has diminished. Only bones and tough connective tissues remain. The cockroaches disperse into crevices and under logs, disappearing into the mosaic of shadowed microhabitats. Their bodies carry no sign of the chemical gauntlet they endured. Their antennae twitch calmly. Their gait is steady. They have walked through rot, fungus and bacterial storms and emerged unchanged. In a world where chemical toxins shape the fate of countless species, the Madagascar hissing cockroach thrives by embracing what others fear. It inhabits environments considered lethal, consumes materials considered dangerous, and transforms toxicity into nourishment. Its survival strategy is not aggression, not venom, not speed, but biochemical resilience. It lives in chemical hell and walks out unharmed. At sunrise on a late summer meadow, when the air still holds the cool clarity of night and the grass bends beneath the weight of dew, a small striped caterpillar clings to a milkweed leaf. The plant towers above it like a fortress of pale green spears. To most insects, milkweed is as dangerous as a chemical minefield. Its sap contains cardiac glycosides, compounds evolved by the plant to ward off herbivores by disrupting cellular function, especially in tissues that control heartbeat. One drop of this milky fluid can send predators into spirals of nausea, disorientation, and in high enough doses, complete cardiac failure. But the monarch caterpillar is built not to avoid these toxins, but to consume them with unwavering appetite. The caterpillar chews a slow arc into the leaf's edge, pausing between bites as droplets of sap bead across the wound. Its body, banded in yellow, white and black, arches rhythmically as it feeds, soft tissues gliding over the leaf surface. Inside its gut, enzymes and transport proteins stand ready to manage the dangerous compounds entering its system. Unlike other insects that succumb to milkweed's defenses, the monarch caterpillar possesses molecular adaptations that protect key cellular machinery from cardiac glycoside interference. Instead of binding to the sodium-potassium pumps of its heart-equivalent tissue, a lethal process in most animals, the toxins pass into storage, accumulating safely within its body. The camera pulls in closer. The sap, bright and opaque, clings to the caterpillar's mandibles. The caterpillar wipes it away on the leaf surface and continues feeding, undeterred. Behind the scenes, its organs sequester the toxins, packing them into tissues that will remain with it through metamorphosis. The monarch does not merely survive the milkweed's chemical assault, it transforms the plant's defenses into its own biological armor. Days pass. The caterpillar grows thicker, sturdier. The toxins accumulate. By the time it forms a chrysalis, its internal chemistry has become a weaponized memory of every leaf it consumed. Inside the jade-green capsule, transformation unfolds. Cells liquefy and reform, reassigning structures into wings, proboscis, compound eyes, and long, delicate legs. And all the while, cardiac glycosides remain locked within the developing butterfly's tissues. Toxicity becomes inheritance. When the chrysalis finally splits, an adult monarch emerges, wings crumpled at first, then slowly expanding as hemolymph flows through their veins. The vibrant pattern begins to reveal itself. Deep orange panels bordered by black lines and spotted with white. This coloration, dazzling to human eyes, is not meant to dazzle. It is a warning, a neon hazard label written across fragile wings. The monarch's beauty is its barrier. Sunlight intensifies as the butterfly rests, warming its wings and allowing flight muscles to gain strength. Then, with a few hesitant beats, it lifts from its perch into the open air. The meadow blooms with asters and goldenrods, their nectar-rich heads visited by bees, hoverflies, and other butterflies. But the monarch's flight is deliberate and elegant, gliding between thermals and adjusting its orientation to maximize visibility. Its orange wings flash like tiny warning flags, turning every movement into a broadcast. Do not touch. The camera switches perspectives, now following a young bird perched on a branch overlooking the meadow. The bird, newly fledged and still assembling its mental map of edible and inedible prey, scans the landscape for movement. A monarch flutters past, slow, conspicuous, vibrantly colored. To an inexperienced predator, these traits suggest that the butterfly is easy to catch and full of nutrients. The bird lunges from its perch in one decisive motion. The monarch attempts to evade, banking sharply, but the bird is faster. It snaps the butterfly between its beak tips and flies back to the branch, ready to consume its prize. The wings beat frantically, scattering orange scales like dust motes. The bird begins to strip away the wings, exposing the soft body beneath. Then the real story begins. The bird's first taste of monarch tissue triggers a reaction almost immediately. Cardiac glycosides bind to receptors in its nervous and muscular systems. The sensation is bitter, vastly more bitter than anything the bird has tasted before. But the chemical effect goes beyond taste. These toxins disrupt ion balance across cardiac cells, causing irregular contractions. Even a small dose is enough to generate nausea and discomfort. Within moments, the bird's eyes widen. It freezes, beak half open, then begins to shake its head vigorously. The butterfly falls from the bird's grasp, wings tattered, but body mostly intact. The bird convulses softly, its digestive system rebelling. In many carefully documented studies, young birds that taste monarchs immediately display signs of distress, retching motions, wing drooping, and a rapid retreat to a sheltered perch. The experience is unforgettable. Days or even weeks later, the same birds refuse to attack orange and black insects of any shape or form. Back on the ground, the monarch rights itself and climbs onto a stem. Its wings tremble from damage but remain functional. It rests, gathering strength, the toxins within its tissues still intact. A predator's memory has been reshaped because of its taste, a form of ecological communication more powerful than sound or movement. The scene expands again, now capturing multiple monarchs drifting through the meadow. Their migration season approaches. Monarchs in some regions travel extraordinary distances, journeying from northern breeding grounds towards southern, overwintering forests. During these migrations, their warning wings become even more essential. Many predators along the route are unfamiliar with the local monarch population, but still learn quickly. One attempt, one bite, one surge of cardiac discomfort, and the lesson becomes permanent. The validity of this chemical warning has been demonstrated in countless experiments. When birds are offered both monarchs and similarly sized butterflies without toxins, their preference shifts immediately after their first encounter. They avoid not only monarchs but also other orange and black species, including mimics that exploit the same visual code. In this way, the monarch's toxicity shapes the evolution of entire ecological communities. Aposematic coloration becomes a shared language across species. Predators read it, prey display it, and survival follows its rules. Back at the milkweed patch, new monarch caterpillars begin their feast. The cycle repeats, plant toxins converted into insect armor, bitter leaves becoming a lifelong shield. In a world where survival strategies range from venom-filled stingers to silk traps and acid sprays, the monarch's weapon is subtle yet devastating. It does not immobilize prey. It does not injure attackers directly. It hijacks physiology from the inside out. As the chapter closes, the camera follows a monarch lifting high into the warm air, its wings glowing like tiny stained glass windows. The breeze carries it upward, away from the meadow, and toward the long horizon of migration. Its orange and black pattern remains stark and unmistakable, a message broadcast across ecosystems. This is not decoration. This is a chemical hazard label, alive and airborne, carried on delicate wings that no predator will soon forget.